Welcome in this small tutorial on DNA damage. In this video, we'll talk about damage produced by ionizing radiation, whether by what we call low ionizing radiation or high ionizing radiation. X-rays are an example of low ionizing radiation. When they travel into biological matter or any matter, they can ionize atoms on their path. This means that electrons of its atoms are ejected and can further travel and cause secondary ionization. With X-rays, these events are widely spaced. The most sensitive target of a cell is its nucleus, that encodes all the genetic information in the form of DNA. DNA can thus be damaged by X-rays, but only a few ionizations will be produced at that location. The story is quite different if we use charged particles, also called ions. Ions interact continuously with the biological matter and produce numerous ionizations along their path. The electrons are ejected in a more dense fashion, and a lot of ionization can occur in DNA. DNA is a double helix constituted of a sugar phosphate backbone here and paired bases. This is your genetic code. When we superimpose DNA and particle tracks, such as here, we clearly see how ions are more detrimental for the preservation of the DNA structure. Produced damage can differ largely. The most frequent are single-strand breaks, double-strand breaks, and base damage. Single-strand breaks means that one side of the double helix is damaged. The cell can most of the time repair this easily on the basis of the other side of the helix. Remember, base is complementarity from your biology class. Base damage is a chemical modification of a base. Here again, because the template is available on the other side of the helix, this is generally not a big issue for the cell. Double-strand breaks are lesions on opposite sides of the helix. They are more difficult to repair because of the absence of template. These lesions are mostly produced by ions. It is possible to visualize double-strand breaks using a technique called immunofluorescence. Here, one can see the nucleus of a cell in blue, and each little green dot is a double-strand break that has to be repaired. Let's compare the DNA damage pattern according to radiation. Here, on the top, these are damages created by gamma rays, which act at the same way as X-rays. Note that the dots are well spread. And here below are damages produced by ions, such as silicon or iron. Because they ionize the biological matter in a more concentrated way, this shows up also on the pattern of double-strand breaks. We can nearly see the ion track itself by the damage it does on its path. Unfortunately, little is known about the long-term effects of such damage on the human body. A lot of this radiation can be found in space. This is why the RISE project is so important and guides us toward a better understanding of damage mitigation that could be transposed to human beings.